Hi, my name is Jamie Foster from the Department of Biomedical Sciences at the University of North Dakota School of Medicine and Health Sciences. And the title of our study is Environmental Lead Exposure and Dopaminergic Dysfunction by myself, Dr. Marilyn Plug, and Dr. Gary Schwartz, all at the University of North Dakota School of Medicine and Health Sciences. The purpose of this study, or the goal of this study, was to translate epidemiologic data implicating lead in water, drinking water, with the risk of Parkinson's disease to an experimental setting. Thus, this research is directly responsive to the TREE, or Translating Research and Epidemiology to Experiment mechanism of the DaCCoTA NIH grant. Some background: epidemiological studies have recently shown the prevalence of Parkinson's disease in US states is positively associated with the quantity of acid rain, where it is hypothesized that the acid rain, or the pH, could mobilize metals such as lead from the soil, increasing its level in drinking water. And this would be in rural, even in rural wells and drinking water. It was also shown that there is a significant positive correlation between the presence of lead in water service lines, and these are the lines which deliver water from the water main out on the street to the residents, our private residents. And there are many of these lead service lines still in use in the U.S., even here in Grand Forks. And the existence of these lines is highly correlated with the prevalence of Parkinson's disease. So the cause of Parkinson's disease in most individuals is unknown, but it involves a degeneration of dopaminergic neurons, and these are the neurons which produce dopamine as their neurotransmitter. And it results in motor deficits and cognitive changes. So this figure on the right here shows lead and acid rain as the lead service lines as a potential source of lead in drinking water. And as I'll talk about, this lead can mimic calcium in activating kinases, which are very instrumental in regulating the dopamine transporter, which is very important in the regulation of dopaminergic signaling. So this disruption, as we hypothesize, may lead to susceptibility to Parkinson's disease. So as I said, dopaminergic neurons release dopamine, and the extraneuronal levels of dopamine are controlled spatially and temporally. This just means the level and time at which downstream neurons are exposed to dopamine is all regulated by the dopamine transporter. So that is shown here. The dopaminergic synapse is shown here. You see that dopamine is stored in these synaptic vesicles, and when this neuron fires, it releases these vesicles fuse to the membrane. It releases dopamine, where they combine to downstream receptors on postsynaptic neurons, and in that binding, that this downstream neuron then fires. So when this dopamine is released, it is taken back up into the presynaptic neuron by the dopamine transporter shown here, and repackaged into these vesicles. All right, and it's very important that this dopamine is repackaged and restored, in that dopamine that is released is prone to oxidation, and this oxidation can cause the dopamine to transform into a neurotoxic form. And so dopamine itself is neurotoxic, and it needs to be sequestered in these vesicles between rounds of excitation, if you will. All right, so this dopamine transporter is very instrumental in controlling this, and post-translational modifications such as phosphorylation and palmitylation of the transporter are what controls its rate of activity, and this is all in turn controlled by an enzyme called protein kinase C, which is controlled by calcium. Now, lead alters calcium homeostasis in cellular mediated processes, it's stored in bone, or can be, so it can build up, levels can build up in bone and stay in a person. And it can mimic calcium in the binding and activating of protein kinase C. An additional study indicated that in C. elegans, which are little tiny worms or nematodes, suggests that lead-induced neurotoxicity in these worms, and they only have a few dopaminergic neurons in these worms, involves the dopamine transporter, and also that PKC deficient worms. So these worms are very easily to manipulate genetically, so you can take out the PKC. These worms that are deficient are resistant to lead toxicity, suggesting that both that and PKC have some role in this toxicity. So together, these studies suggest to us that chronic PKC activation, which results in that phosphorylation and decreased that palmitylation, may be influenced by lead and result in altered dopamine homeostasis and neuronal degradation. So that's basically our hypotheses going into this. And then the lead being provided in the drinking water may result in altering that function and dopamine homeostasis, leading to neuronal degradation. So because of funding delays and then delays in the hiring process and identifying somebody to hire to do these studies, we have just gotten started just recently. And initially we just did an acute study with cells, kidney epithelial cells. So this is not a neuronal system, but we are moving into neuronal systems in animals here. We're in the process of doing that. But in this little study, we just used between 0 and 500 micromolar lead acetate. And as a control, we used sodium acetate and exposed these cells in buffer medium with these two compounds from, as I said, 0 to 500 micromolar, just to see if lead had any acute effect on dopamine transport activity. And as we can see here, there's a trend actually in an increase in activity, but this was not statistically significant. So basically, there was no effect of lead on dopamine uptake, at least in an acute setting in a non-neuronal system. And so we are in the process of switching to a neuronal model system and doing more chronic exposures over a month with an animal model system. But our conclusions so far are that the 2-hour exposure with up to 500 micromolar lead had no effect on dopamine uptake in LLCPK kidney epithelial cells expressing that, suggesting that this model system may be resistant to the deleterious effects of lead, or greater times of exposure are necessary for effects on uptake to be seen in this model system. Follow-up studies, as I said, in a neuronal model system are underway, and this model system may be more sensitive to the effects of lead, because in general, neurons are a little bit more delicate than these epithelial kidney cells for certain. Long-term 30-day exposure studies are also underway, where animals will be exposed to lower and a higher concentration, 30 parts per million or 1,000 parts per million of lead in their drinking water over a 30-day period or 1 month. And we anticipate this chronic exposure will lead to altered dopamine uptake, and that palmitylation and phosphorylation status will possibly also be altered in striatal synaptosomes isolated from these animals. So that is the summary of what we have so far, and we hope that we will be able to complete in the next month or two these other neuronal model systems to see if it supports our hypothesis. Thank you for your time.